Hello, everyone, and welcome to this third series for the African Plant Breeders Association and the African Bioinformatics Institute on statistical genetics for agricultural and agri-informatics. We are very lucky to have Dr. Réka, Professor Réka, Associate Professor Réka, with us from the University of Nebraska-Lincoln. Associated Professor Réka Howard is a professor of statistics and the director of consulting in the Department of Statistics at the University of Nebraska-Lincoln. She's also a courtesy professor in the Department of Agronomy and Horticulture at the University of Nebraska-Lincoln.
ايوه هجمته النور.
A couple of housekeeping rules. Please keep your mic on mute, and if you've got any questions, we'll take them during the lecture. You can raise your hands or you can type them in the chat, and we can also take them after the lecture as well. So welcome to this third lecture, and over to you, Dr. Réka.
Thank you very much, Sumir. Thank you for the introduction, and thanks everybody for joining this lecture. So this is the third lecture that we are having. And so today's idea is that we will cover some of the blocking aspects and repeated measures. So I just have to announce two things. So one is unrelated, that several of you emailed me about design-related issues. I think I emailed back everybody. If I did not email you back, it means that I did not see your email. So please email me again. And the other related announcement is that there are a lot of aspects that we are not covering in this lecture just because of the time constraint. So we have not even an hour to cover two important aspects in experimental design. And I just want to emphasize that there are a lot more to it than what we will cover. So, but in general, I want to talk about blocking first and why we are doing blocking and what's blocking and how do we do that. So far in our previous lectures, we talked about completely randomized design, and we mentioned blocking, but we didn't really go into it. So as you remember, we talked about factorial design, and the arrangement was CRD, so completely randomized design, where what we do is we have treatments, and the treatments have equal chance of being assigned to more or less favorable material. And in this case, some treatment will be assigned to more favorable material by chance. And we want to eliminate that not eliminate, but reduce the chance of that some treatments will be assigned to more favorable material. So what we do is so-called blocking. So what we do is we will assign our experimental units into these blocks or groups. So we will be grouping them. So I listed some ideas for blocking. So one thing to remember that in experimental design in agriculture, many times our blocks are actually locations or parts of the field, but they can be days, they can be operators, and actually they can be also continuous. So they can be light, they can be temperature, they can be moisture, and I mean it in a continuous way. So not no light and light, but we would specify the amount of light that, let's say, plants receive. So what we do is we assign the treatment in each block. And so what we will talk about is randomized complete block design. So there are many ways to block, but what we do is we will make sure in this lecture that each treatment is assigned in each block once. So what we will do is we will have treatments, and each treatment will receive an equal allocation of favorable material. And so it's very important that we can actually block correctly and some way that helps our design, and we can actually block incorrectly and it's hurting our design. So we want to make sure that we are blocking effectively, and we will talk about what does it mean to block effectively. So the idea behind blocking is, so we are dividing the experimental material into homogeneous set of units and apply a set of treatments to these homogeneous units. So what does it mean? So we will divide the experimental units and we will group them. And by this, we want to eliminate some variation that's not due to the treatment, but potentially due to the field that is not due to the treatment. And one other important aspect that we don't necessarily emphasize, but it's important, that the blocking also allows us to include a wide variety of experimental units, so we can actually increase the inference space of our study, because we will have, let's say, more fields, so not just one field. So if we are running an experiment and it's on one field, then the inference space is of our study is that field. So we cannot conclude anything on other fields. So this is a summary, and this is very, very important. I would say this is the key of how we need to block. So we want to organize experimental units into groups in such a way that between the blocks, we want to have the second one. Between the blocks, we want to make the experimental units as different as possible, and within a block, we want to make sure that they are as homogeneous as possible. So there are a lot of repeats in the notes, but this is the key, so I won't go through everything because otherwise we won't be able to go through the whole material. So there is this description how we can actually write blocking into our model. So I already mentioned that we'll be talking about the randomized complete block design, which is RCBD, and it means that we are having one experimental unit assigned to each treatment for each block. Because actually what we could do within a block, we could have replication too, but that's another design, meaning it's not more complicated or not much more complicated. We just have to account for the replication too. But we are not talking about that. We're just talking about the blocking aspect. That's why we are talking about the most simple one. Also, we could talk about incomplete blocking, where that's actually a more realistic scenario for many researchers, because you will have some missing data. But the good news is that you will analyze those in a similar way than randomized complete block design. But one thing too about incompleteness, that if you are actually planning your incomplete block design, the best way is to have it as balanced as possible. So this is the model. We saw a model similar to this. Just a reminder, it doesn't matter which letters you use. You could use alpha, beta. You just have to know what letter stands for what. So in this case, we are having a response. We are having an overall mean, which is the mu. This tau I stands for the treatment effect. So again, this could be a factorial. So this could be alpha, beta, gamma, and the interactions and so forth. But now I'm just denoting one treatment effect and then one blocking effect. That's another thing too, that there is a row column design, for example. Maybe you heard about that. That just means that we are having two blocking factors. So in this case, we keep it simple. We are having one blocking factor because just we are talking about the idea of blocking. So in this model, this beta J stands for the blocking. And then we are having the regular error term that's assumed to be normally distributed with mean zero and sigma squared variance. In the notes, if you see something like this, which is like a rectangle, it's just a weird error here. So this should be a sign for distributed as. So one big question is, and we will talk about it in a minute, whether this should be random or fixed. And that always comes up when we are designing an experiment, whether the blocks should be treated random or fixed. fixed, and we will talk about it, and we will talk about what the difference is between random and fixed. But first, let's just try to picture very quick example of blocking, where we are saying that we want to evaluate the effect of elevated carbon dioxide on rice grain yield, and then we are having 4 blocks. So the blocks would be farms. So which means that different fertilizer amounts, different management practices, and then in each block we are having 2 paddies. So those will be the 2 treatments. So just imagine 4 farms, and within the 4 farms you are having 2 different paddies, and the 2 different paddies are receiving the 2 different treatments. So this is how we could just picture a block design. So one thing to go back to this model, one thing to notice, I did not write interaction between the treatment and the block. So we assume, and this is an assumption, that there is no interaction between the treatment and the block, because they should be independent. That's the whole point. But we can check, and it's a good idea to check whether there is an interaction between the block and the treatment. So in this example, so let's say we are having 4 blocks, this rice example, and then let's say that this is a data set that we got. We plot this and we say, oh, okay, I have the treatment on the x-axis, and then I have the response on the y, and then I'm plotting the different blocks, so the 4 blocks. So there is no obvious interaction, so we are fine. So that's another topic, how to deal with interaction in this case, but we are not going into that. So one thing to realize, I mentioned this last time, there is a treatment design and an experimental design. So both of them are important, and we have to talk about when we are designing and analyzing a data set with experimental design. So so far, or before this lecture, we talked about the CRD, so the completely randomized design, and that was on experimental design, meaning we talked about the layout of the design. So this RCBD is just another layout. So with the layout, so the blocking, we have to pair this with a treatment design. So a treatment design could be just a one treatment design. So in this case, actually, we have that. This is that example. So we have having 3 blocks. And then within a block, so block 1, we are having 4 treatment levels. So control and treatment 2. So pretty much 4 treatments. And then in block 2, also we have the 4 treatments. Very, very important that you need randomization, and you need different randomization in each block. That's very important. But there can be other treatment design. So we could have actually regression. So you can see that this is, again, we have 3 blocks, and we are having a quantitative factor. So whatever that is, with numbers 20, 60, 80, 40. So these are randomized, and they are randomized in each block separately. And then we can have also factorial. So we can pretty much put together the factorial with blocking, and we can do whatever we want. Just keep in mind, we have to talk about experimental design and treatment design. So we already talked about how to choose the blocking. That's very important to maximize the difference between blocks and minimize the difference within blocks. So this is just a little example that how do we want to block. Maybe I will skip this just because of time. And then the question is how large the block should be. Many times it's already, so if you're designing an experiment, you don't really want a huge number of blocks because it's very, I'm sorry, not a huge number of blocks, but the number of treatments within the block, because then it's very hard to make that block homogeneous. So that's one thing that we have to keep in mind. We have to remember the no block by treatment interaction. Sometimes we have that and we have to deal with it. That's a different topic. These are just, again, just different examples for blocking. And then the very, very important other question is whether it should be fixed or random. And it comes up all over, like a lot, that whether the blocks should be random or fixed. And the answer is it depends. So when do we choose them fixed and when do we choose them random? So if these blocks are just samples of a population of blocks. What does it mean? So let's say that there is an institute and the institute has farms and they are testing varieties and they are having a lot of experiments. So let's say that, just making up something, this institute has 1,000 farms and you randomly choose this is a bad example, sorry, not 1,000. Let's say that they have 50 farms in one area of a state. And then you choose 5 of those blocks. Then, meaning fields, then you would consider those fields probably as random because those blocks are just a random sample or random samples of the population of blocks. But let's say that you are a farmer and you have 3 farms and you are evaluating your varieties and you are running your experiments on those 3 farms. Then you are really interested in those farms, so that's when you will choose them as fixed. Because when you choose them as fixed blocks, you will get the mean estimates. So you're focusing on the comparison between blocks. Even though a block is not a treatment, so you have to be very, very careful. With the random blocks, you will get variance component estimates. So different approach, but one other very important thing to consider when you have balanced data, so let's say that everything is beautiful and not missing, then it doesn't matter if you are choosing them fixed or random because your results will be the same. On purpose, I did not include data, and I did not want to include too much of a syntax for code because it depends on what software you are using. So that's not the point here. If you need help with actually implementing it, let me know. But here I'm just focusing on showing you how you would do this in general in a software package. So the important part is that you can have the block random or fixed. So you have to tell the software that it's either in a classification statement. So in this case, I have to say class block. So I have to tell the software that whether this is it will be a classification variable, but whether it's random or fixed. So in this case, since I'm not assigning the block to a random statement, so then I know that it's fixed, and then here it's random. Again, don't worry about the syntax, just whatever package you are using, there worry about whether you're specifying your block as fixed or random. So we have probably I need to, I'm just thinking about that we have a lot more to cover, and maybe I am just skipping some of these just to make sure that we get to all of the important aspects. So I will be skipping that example. The only thing that I will just show you that when you are having a block, so in this example, and don't worry about the example, I specify a block as rep, that's the name. So it could be block 2, it could be pumpkin, it could be whatever I say. And then we can see p-values, and this chem So the chemigation is my treatment. And that's what I'm interested in. What I want to point out, and don't worry about the example, that you will get a p-value associated with the block. And many times we get the question, should I block or not? So this p-value associated with the blocking won't actually answer that question. And I just wanted to say that sometimes this will be not significant like here. That does not mean that you should not block. So we will talk about later how to actually determine whether you should block or you should include the block in your analysis or not. So that was the only thing I wanted to show here. And then you can get estimates and depending on your um, um, example and your software. So I am skipping this example. So, but one thing that I will mention again, blocking has to be paired with a treatment design. So in this case, I did not talk about the example. We had one treatment factor with several levels, and those levels were qualitative. We could also have quantitative levels. So there is another example here where you can see actual numbers, 0, 50, 100, 150, 200. So what we do is then we combine the blocking with regression. So the regression was when we talked about the idea of factorial design, and we had a qualitative by quantitative example or quantitative by quantitative example. So what we have here, since we are having 5 levels and it's regression, we can have up to 4 levels in the model. So what it means, we can 4 levels or 4 effects in the model. What it means, I can add the linear effect, the quadratic, the cubic, and the chordic, and that's it. So I can add 4. I know that it depends on your software, but I just left this syntax in to show that. So this was phosphate. So the phosphate levels, and then so I am adding the linear, the quadratic, the cubic, and the chordic. And then what we do is, I mentioned with the factorial design that we have to tell the software to use the type 1 sum of squares because it's regression. That's important too. But also look that there is a block included in the model. So it's exactly the same thing what you would do, for example, what we did in our second lecture, but we are adding a block effect to what we did. So that's why we are having the combination of a treatment design and an experimental design. And then again, we are getting output. And for the regression, I mentioned last time that what we do is we look at what terms are significant. So here, just the linear effect is significant. So we rerun the model with the significant term, and that's our final model. So that's what's happening here in the output. Don't worry about output. I just want to emphasize ideas, what you have to pay attention on. So there are a lot of details here, how to use the output. I'm more than happy to share the notes. And then if you have questions later, I know that I'm not covering a lot of things, but I left them here on purpose because if you are interested, I'm happy to help to figure out how to use this. So this is, I'm just going through outputs, and I'm not really mentioning anything about these because again, you can go through if you wish, and I can help you with that. So one thing here is why we have so much output. So in case you will look at it later, because sometimes I treated the block as random, and then first what we do here. So here too, it's actually random because we are running the whole model. But before that, we treated the block as fixed, and then it's just describing how the output will be different when it's fixed and random. And then you will actually realize that in a balanced case, we are getting similar things, but whether you are getting variance component estimate for the block or not. So when we are having a random block, you will get a block estimate for so this is our variance component estimate for the block. But your conclusion will be very similar in terms of your treatment. So I mentioned that, and that's very important too, to efficiently block. So blocking that doesn't just mean that I will add one more term into the model. We have to think it through. So I mentioned last time, but I will mention this time too, that, for example, when you have a field, and let's say that one part of the field is flooding and the other part, so let's say that you are having a rectangle field, and then on the left side, so my left side, so let's say one side, left side is flooding and the other side is sunny. So you want to make sure that you block that way, so like this way, so all of the flooded part is one block and all of the sunny part is one block, not the opposite way, so not left to right in this way, because then in one block you would have flooded and sunny, and in another block you would have flooded and sunny. So you want to make sure that one block is as homogeneous as possible, but when you compare the blocks, they are different. So just think about it. In one block, I want to have flooded, and the other block I want to have sunny, not differently. But how do we actually evaluate whether we should be blocking or not, other than knowing these things that I know something about my field, so I know how to block? We can calculate so-called efficiency, and there are other ways to do this too, but this is very, very simple. So what we calculate, and it's very simple. That's like you can complicate your life, but you don't have to. So what you do is we want to minimize the error variance. That's what we want to do, and we want to account for the variability in these blocks and elsewhere. So we calculate the mean squared error. So this is the error associated or the variability associated with the error for a design where we just take the block out versus we put the block into the model. So what we do is, so we are having CRD, so which means completely randomized design. So we are not blocking. So we are ignoring the blocks. We run that design, and then we are getting this, it's so-called sigma squared CRD, which is an MSE. So the variability associated with the error. So this is the residual variance estimate. So you get that, and then you also get the same thing, so this error variance for an RCBD, so for the design where you are including the blocking. So this is just describing the variance estimate associated with estimating the difference between two means. So, and then these are the formulas. Do you have to remember? Absolutely not. Just remember that actually the relative efficiencies that you want to compare these two things, because then what we want to do is we want to minimize the error variance. So we are calculating the estimate of these error variances based on blocking, no blocking. So this is the blocking and this is the no blocking. And then if you are calculating it this way, then the relative efficiency, if it's greater than 1, what does it mean? It means that this part is greater, so the error associated with the CRD is greater, and then then the precision for the RCBD will be higher because you want to minimize. So if you don't even remember this, that's fine. Just remember that, okay, I want to compare the two error variances and which is smaller. I want to have the design where my error variance is smaller because I want the block to account for variability. So this is just another more complex formula for the same thing where we are adjusting for the degrees of freedom. Don't worry about it. I would not remember this formula, and that's not the point. It's just roughly speaking, you want to see whether you are blocking correctly or not. So this is just an example how to actually do this. So either including or not including a block, getting the output, and then calculating the efficiency. So I am skipping that. So what I want to do, since it's already in my time 8:30, so I want to jump to repeated measures. So far we talked about blocking, and now we are talking about repeated measures data. Why is it important? Because many times what we do is we collect data and we measure the response over time on the same experimental unit. It's very important that we are collecting the information, so we are measuring the response on the same experimental unit. So why do we do this? Because we want to, for example, ensure that the treatment is effective over a specified period. We want to monitor safety over a specified period, or we want to know when some treatment becomes effective. So bottom line, still, we will have treatments. We are looking at the response over time. So we want to look at I will just show you the model. We want to look at a treat. So here alpha will be a treatment effect. Beta can be the time effect. We can look at the interaction effect. And then we have the error terms. So when you look at this equation, so this model equation, you will say, oh, I saw this before. It looks like a factorial. So why do we have to even bother with these repeated measures? So yes, the model equation looks like a factorial because we are having one factor, another factor, which will be here the time, so that can be another factor, and then we are looking at the interaction between the two factors, here the treatment and the time, and I have an error. So that's the key. The error is actually different than for a factorial, because for a factorial design, we just said, so actually we could have a factorial design here combined with repeated measures, just saying. But the error structure is different because when we just had a factorial design alone, what we assumed that the error was homogeneous, so had a homogeneous variance. So what does it mean? That no matter which treatment I'm talking about, the assumption is that it will have the same variability. And then now, so one thing that we have to realize that what do we do when we have a factorial treatment design? We are assigning the treatments randomly to the experimental unit. But here, the time is not randomly assigned. That's fixed. So when we have one experimental unit and we are measuring the response on that experimental unit repeatedly, those measurements on the same experimental unit will be correlated because we are talking about the same experimental unit. So this is just describing one example. Don't worry about it, but it's in the notes so you can go through. But the important aspect is that two measures on the same subject will be usually be positively correlated. And then two measures close in time on the same subject will be likely be more highly correlated than two measures made further apart. Not always, but these are the general assumptions. And then it depends on the covariance structure, how we are actually describing these correlations. So again, this thing won't be simple anymore, this term, this error structure. So what happens here? So we are having this structure that can be described in a matrix. Actually, when we had the homogeneous error structure, we can describe that in a matrix too. It's not the matrix part that's special. It's the special part is how this matrix is set up. So what's this R1, R2, R3, R4? So what it is, we are having in this case 4 subjects. So this is just an example. Let's say we are having 4 subjects. And then within a subject, so subject 1, I will have this submatrix that will describe the measurements and the correlation between measurements. So what we will see, I will show you more examples here, but just imagine that this R1 is a little submatrix, meaning this is a matrix, and then this R1 can be another little matrix, and then R2 is a matrix, R3, R4. They don't have to have the same dimension. And why are these zeros? Because still we are assuming that the subjects are uncorrelated. So R1 with R2 is uncorrelated, so that will be a matrix, a submatrix with full of zeros. So let's talk about covariance patterns and how do we do this. So one very, very good news is that you don't have to code any of this because everything will be included in your software package. So depending on which software you are using, different covariance patterns are included. So actually I use SAS for this, and SAS is very organized and has a lot of covariance patterns. R is actually less organized because some packages would have some covariance patterns and other packages would have other covariance patterns. That doesn't mean that you cannot do that, and that it doesn't mean that you have to have all of the covariance patterns available, because you can have a few and you are fine. So what are these covariance patterns and how do we choose? So the thing is, the first thing which I tried to emphasize last time too, before you run any model, you want to understand your data. You visualize, you look at it, you calculate summary statistics, so you have some sense what's going on. So that will really help determining which covariance pattern you want to use. So there are several ones. So we will consider a little mini example where we will say, so instead of these 4 subjects, I will just say I have 2 subjects, okay? And let's say that subject 1 has 3 measurements. It says visits, but 3 measurements, and subject 2 has 4 measurements. And what we do is we will look at different covariance patterns. Maybe we won't have time for all of the covariance patterns, but I will just show you a few. But there are several ones listed in these notes. So the first one is it's called independent covariance pattern. So let's look at it. So this is the matrix describing this variance covariance matrix. So what you can do is imagine that this is my R1 submatrix, okay? And then this part here is my R2 submatrix. Just remember, we had 2 subjects, so we will have R1, R2. So I have this 0 submatrix, and then I have this other 0 submatrix. So the first R1, so the R1 submatrix is a 3 by 3 matrix here, and the R2 is a 4 by 4. Why is that? Because we said that the first subject had 3 visits or measurements, so that's why it's a 3 by 3. And then this one is a 4 by 4 because we said that it had 4 visits or 4 measurements. So we have 2 subjects. First one is receiving 3. The second one is receiving 4 measurements. And then it's called independent because so we don't assume homogeneous variance anymore, because if we would assume homogeneous variance, it would mean that the sigma 1 squared, sigma 2 squared, sigma 3 squared, so we would just have sigma squared. So everything would be the same. So these would be the same, but they are different now. So I have a sigma 1 squared, sigma 2 squared, sigma 3 squared. Oh, sigma 1 squared, 2 squared, 3 squared, 4 squared. Oh, what's going on? So the variance associated with the first measurement is sigma 1 squared, with the second one sigma 2 squared, with the third measurement sigma 3 squared. Same here, first measurement, second, third, but the second object has or subject has 4 measurements, so that's why we are having sigma 4 squared here. So these variances are different now. So that's why it's different than homogeneous variance, but it's called independent because in the off diagonal of the submatrices, we have zeros. What does it mean? It means that we are assuming, so this is an assumption that we are making, that the measurements are uncorrelated. So the zero covariance between measurement 1 and 2, 1 and 3, and then 2 and 3. So this 0 represents the covariance between the measurement 2 and 3. So that's why we have a lot of zeros. This is just saying like, okay, this is what I assume. And then there are other ones. So this is called the general or unstructured covariance pattern. So here, actually, we are assuming everything is different. So again, we are having these two submatrices. Still, we are assuming two subjects, three measurements for the first subject, and four measurements for the second subject. But now notice that I have sigma 1 squared, sigma 2 squared, sigma 3 squared in the diagonal. Again, sigma 1, 2, 3, 4 squared. So that was the same for the independent covariance structure. But now in the off diagonal, we are having these thetas. So I have theta 1, 2, 1, 3, 2, 3, which means I am assuming different covariances depending on which two measurements I'm talking about, because this theta 1, 2 is the same as this theta 1, 2. But keep in mind, this is much more complicated than this, because here we just assume that those are zero, and now we are assuming that they are different, and those are parameters that we have to estimate. So it's when we are complicating our lives and we are including more and more parameters, it gets harder and harder to estimate because we have more and more parameters to estimate, and we need the sufficient data for that. But this is one covariance structure we can assume. There are other ones. So there is this called AR1 or first order autoregressive covariance pattern. What does it mean? So actually here I am multiplying the whole matrix by sigma squared, which means sigma squared times 1 is sigma squared. Sigma squared times rho, this is a rho, is sigma squared rho. So every element is multiplied by this. So in the diagonal, I have that homogeneous variance, but I have off-diagonal elements that are not zero, and this is the assumption that when I am one measurement apart, the covariance is sigma squared rho. When I am two measurements apart, it's sigma squared rho. Sigma squared rho squared. So that's how I distinguish between how many measurements apart we are. So this is just, again, another covariance structure, and still we are scratching our head. How do I choose? So one thing is you look at the data and then you see, oh, okay, how do the correlations change between measurements in my data set? What can I assume? And then maybe you say, I have no idea how to figure this out. So good news is you can actually use a so-called information criteria to decide which covariance pattern to use. So I will just show you one more covariance pattern because this is very important. It's called the compound symmetry, and what it assumes, homogeneous variance and same covariance no matter which two measurements we are talking about. So it's theta, theta, so everywhere theta. So this is called the compound symmetry. Why is it important? We didn't talk about split plot design, I understand, but this is also called the split plot in time. So actually, this can be also modeled with a split plot design where the split plot factor is time. Uh, don't worry, we didn't talk about split plot design, but if it means anything to you, that's how you can connect compound symmetry to split plot design. So I won't cover these covariance structures. There are more, actually, there are more covariance structures than these. And then you can actually code your covariance structure, which you do not want to, because you don't need to. These are fine. And then I mentioned this information criteria. So what you do is you run your model, and then you will see an AIC, potentially an AICC, and the BIC value. So for sure, no matter which package you are using, you will see the AIC, but I believe you will see all of these three. You can pick your favorite. AIC is a good one too. And then you compare the AIC values for the different covariance structures. So what you do, you run your model with one covariance structure, you look at the AIC value, and then you run the same model with a different covariance structure, and then you look at the AIC. So you want to minimize the AIC values that will have the best model fit that among the models that you are choosing that has the smallest either AIC, AICC, or BIC values. Don't mix AIC with AICC because they are calculated differently. Don't mix AICC with BICC. So pick one and then choose the model based on that information criteria. Many times what we do is we look at all of them and then just make sure that all of them are giving the same message, meaning they would choose the same covariance structure. Sometimes it does not happen. Sometimes actually they choose different ones. That's when it comes into a picture that you look at your data and then which covariance structure makes sense. Because when we looked at these covariance patterns, you have to actually look at them and say, okay, does it make sense that I am assuming that the covariance is, for example, are the same. This one, the compound symmetry, or it doesn't make sense that I'm assuming that. So you have to spend a little bit of time to figure out which covariance pattern, but it's relatively easy. So I really wanted to talk about this part. So I ended up leaving code here. Again, the likelihood that you are using SAS is really small. It works differently in R and other software, but I just wanted to leave something here. So I left the code here and I left the output. And just to show you that this is how it looks, that for one model fit, you would get the AIC, ICC, BIC values. And then you don't compare these with each other. What you do is you run another model and then you would compare, for example, an AIC here with the AIC here. Actually, those are the same thing because I ran the split plotting time and compound symmetry, which are the same thing, just coded differently. Don't worry about it. But this is how you compare AIC values and determine which covariance structure to use. So this is one important thing, very important, that I did not mention, that for this example, I assumed that the measurements were equally spaced. So if the measurements are not equally spaced, so let's say that you measured your response at week 1, and after that week 2, and after that not week 3 but week 4, and then week 7, then there are certain covariance structures that you can use, and there are certain ones that you should not use. So, for example, the AR1 is one that frequently used for that scenario. There is one that's not even included here that's called spatial power. That's commonly used for that scenario, and there are some other ones too that you can use. But important fact that if you are having unequally spaced time intervals, be careful which covariance structures you are assuming to use. So this is what I have. I know that I rushed through a lot of things. I want to emphasize important aspects of blocking and repeated measures. We did not have time to implement anything, as we did not have time to implement anything for the factorial design either. I'm very happy to help if you need help with any of this and figuring out notes and just if you have any questions. I think I will stop talking here, and then if you have any questions, please let me know.
Great, thanks so much for giving us another really amazing lecture. It was very insightful. I hope that the folks on the call have learned quite a lot. Are there any questions from the group? You are welcome to type them into the chat, or you can raise your hand if you'd like. There was a question that came in a little bit earlier from Asimbuyu. Asimbuyu, do you want to unmute and just talk to your question?
Okay, thanks. I don't know if she can see the diagram I sent.
No, I just see some text.
Okay, let me just describe it. So there are three different varieties being tested at different fertilizer rates. So 4 rates, 100%, 75, 50%, and 100. So to make it easier, sorry for the background noise, to make it easier to apply the fertilizer, all the 100% rates were put in one block running north to south, then 75, then 50, then 0. So later I was told that that doesn't make it an RCBD because the fertilizer blocks are all in the same position. All the hundreds are to the west, all the zeros are to the east. So I was wondering whether it justifies trying to avoid human error to keep all the fertilizer rates in the same place. So it's easy to apply. The person knows I'm using the 100% container now. I'm using the 75. And also to avoid runoff between the plots. If 100 is next to a 0, what if some fertilizer runs into the next plot? That was the question.
Okay, so do I understand it correctly that so you are having 3 fertilizer levels and you don't have replication, is that correct? So you have a chunk of
There are four replications.
so you have
the reps are on top of each other.
Okay.
North to south.
Okay. And then so I'm sorry, what's the question?
Okay, it's
Oh,
showing the
perfect.
pictures
I
there.
can see. Okay, perfect. I can see.
So all the 100% plots are the ones that start with 1.
Uh-huh.
And those that start with 2, 75%, 3, 50%, then 4, there was no fertilizer added.
Okay.
So this plan was so that the one applying fertilizer doesn't make an error. They know that when I'm in these plots, I'm using this container. The next one, I change the container and just like that. So the reps are running like from top to bottom. Rep 1, 2, 3, 4.
Okay.
So we advise that this isn't RCBD because all the hundreds are on one side, all the 400s are on the other side.
Yeah. Yes. Just a second. And then you are having so how are okay, you have the 3 varieties. Is that correct?
The varieties are randomized. That's the different colors, like the maroon, purple, blue are the varieties.
So you are having some split plot here.
Okay.
Because you are splitting your fertilizer field, and then you are having the different varieties. Yes. So
that's what we need to clarify. So is it not a good thing to do?
No, it's fine. No, it's fine. It's fine. It's just you have to model it correctly. So I would model this with a split plot. No, this is a very general look, like, yeah, it's fine.
Okay,
And you are
thanks.
having replication. So actually you are having the reps. So your reps are the so-called blocks.
Okay, thank you.
Oh, no problem.
Great. Are there any other questions? I'm just checking the chat. I don't think there are. I don't see any others. I also don't see any hands raised. So I can give it a second or two in case you want to either type your question, or you want to unmute and ask your question?
And please keep in mind, if you have questions later, don't hesitate to email me. And I'm happy to meet with you and discuss. And yeah, so.
Okay, we have one hand that's just popped up. So, Shadrach, do you want to go ahead?
Yes, thank you. I was just following on as you assume this question. I'm seeing that the fertilizer application is ordered, and the plots within the plots, there's a subplot of within the subplot, the fertilizer application is ordered. Is that I was just trying to follow and wondering how that would reflect in the analysis or create biases.
So what do you mean by so we have there they have the
There is
fertilizer, and then within the fertilizer they are having the three varieties. So those are the sub plots pretty much. So and then you have so there are three things going on here. So you are having the reps, you are having the fertilizer, the four levels of the fertilizer, and then the three levels of the variety or three varieties. So you want to have the reps as kind of like blocks, and then you are
the
having
varieties are four.
What's that? Sorry.
There are three varieties.
Three
Four
varieties. Okay,
fertilizers.
now.
Yes. Yes. So four blocks, four fertilizers, and then three varieties. So
Okay, sorry, I
Oh, it's okay.
understand.
And then, so what you have is you are dividing the fertilizer blocks for the varieties. So so the fertilizer is the main plot, and the variety is the subplot factor in the split plot. Does it make sense?
Yeah, it makes sense. I thought that the varieties were four, so it's okay.
Any other question about this or anything else?
There's a question from Lydia on the chat. She's just asking, about statistical software and programs, how to access some programs and software. It's always a tricky question.
So, yes. So, there are a lot of different software packages and the software that you can use for these. So what I have in the notes is actually so-called SAS code that's there is an academic version that's free, but the standalone version is not free. For a lot of analysis, the academic, so if you are in academia, you can access the academic version. Um, the other thing is I have a lot of this is in R. So R is freely available. So if you need anything related to these, so those are the two things I can help with, SAS and R. But that does not mean that you have to use SAS or R. If you are already have your favorite packages, then please stick to that, because it's much easier to keep it that way than learning something else. But yes, so if you need any specific example, please email me, and then I can share examples. I can share in SAS and I can share in R.
Perfect. And then Kwame also has his hand raised. Kwame, do you want to go ahead?
Thank you very much for the opportunity. I just wanted to refer to the last example. Maybe I missed something. If it is possible, if they can put it up again. But I just wanted to ask if the main plots shouldn't be randomized also within each replication.
The main plot randomized within the
Each
replication.
Sorry
replication.
Yes. You want to do that. Like you don't want to have the same order. Yes.
Because I thought from what they said, it seemed like a certain
Should.
fertilizer was just on one side
Yes.
throughout all the replications, that I don't think that should be.
Yes, should, but sometimes you just can't. Yes. Theoretically, you
Okay.
should. Yes, you're correct.
All
But
right.
there are some logistical issues that you just can't.
I know, I
That would be
understand. I
the
just
best
wanted
way.
to check. All right.
Mhm.
No, thank you.
Oh, no problem. A lot of times there is some randomization kind of missing, and then they have to be aware of that too, that maybe there is some issue there, that it's not just due to the fertilizer, but the side of the field. But many times it's not easy to apply certain treatments truly randomly.
Okay,
So,
thank
but
you.
they actually had more randomization than some other one. Like this was better than could be.
Great. Thanks for that. Then, okay, I don't also see any other questions on the chat. Just maybe a note from Sumir that we will have our next lecturer start the next module, and we'll be releasing a lot of that information soon. Sumir has already put some of the reading materials for the next session, as well as some of the registration links on the chat, but we will make sure to disseminate that widely again, similarly to these sets of lectures. And we are also making sure to record all of the lectures and make them available either on YouTube or via our website. So you will be able to get them from there if you have missed anything or you'd like to review anything. And you are welcome to get in touch with us if you'd like us to connect you with Dr./Professor Howard. We are more than happy to do that. So yeah, I've waffled on a little bit and no other hands have come up and no questions on the chat. So I think this is a good point to end this particular session. Just again to Dr. Réka Howard, just thank you very much for making time and for also just being very patient with all the questions and taking your time to answer all the questions very thoroughly. I think everybody on the call and everybody that's attended over the last couple of sessions, I've really appreciated your patience and your willingness to share your knowledge and to help guide some of the people on the call with some of the current research design questions, which I think has been incredibly useful and helpful. So yeah, Dr. Réka, I don't know if you just want to just close off the session there for us, and then yeah, that's a wrap.
Thank you very much. And thanks everybody to join. And I really feel bad that we didn't discuss many details. So if something is not clear or you want to learn more about something, please don't hesitate to email me, because I'm happy to talk more about these things. So anytime. And the only thing is I mentioned my last week travel. So next week I will be traveling again all week. So if you email me and I don't answer until the following week, which is the week of the 29th, forgive me, but I will be pretty much be able to answer. If you email me now, I will be able to email until Thursday, and then for a week I won't be able to answer, but then I get back, I will answer your email. So thank you very much.
Thank you so much, and thank you to everybody on the call.